This figure is showing us the process of DNA sequencing using a dideoxynucleotide. So let's just stop for a moment and think about. If we're going to do a DNA sequencing reaction, we're going to need a couple things. The first thing we're going to need is the template DNA that we want to sequence. The second thing we're going to need is a DNA polymerase molecule because we're going to be making new DNA. The third thing we're going to need is DNTPs, in other words, A's, T's, G's, and C's, in order to use in making the new DNA. And the fourth thing we're going to need are DDNTPs. And we're going to need four different types of DDNTPs: the DDA, the DDT, the DDC, and the DDG nucleotides. And we're going to add some buffers and salts to this, and then we're going to start the process of making new DNA, but allowing termination to occur as well. Now you have to remember, in the tube where the sequencing reaction is taking place, you don't just have one copy of the DNA template. You have lots and lots and lots of copies. So please remember, we're only showing you one of the possible copies in this particular figure. So the first thing that has to happen for a sequencing reaction to occur is you have to denature the strands, and this is done in a very similar way to PCR using heat. The second thing you have to have is a primer. Just as we did in PCR, we have to have a primer because remember, DNA polymerase requires a primer to extend the growing nucleotide strand. Now, naturally occurring, this is the function of primase. However, in a sequencing reaction, we actually put in a short synthetic strand of DNA that's complementary to the end of the region that we want to sequence. So that primer, when we cool the reaction down, the primer is going to find its complement by the process of hydrogen bonding, and then my DNA polymerase, and Taq is just one type of DNA polymerase, is going to start adding the DNTPs, because remember, those are present as well. Now, what you have to remember is in the reaction tube there are the 4 DNTPs, but there are also the 4 DDNTPs. And what you do is you put the DDNTPs in the reaction in very low concentration, so that probability would dictate the odds are a normal nucleotide will get inserted. But because of the small concentration of the DDNTPs, there is the probability that a dideoxynucleotide would be inserted for any particular nucleotide as we're elongating this chain. And so that's the basis of the DNA sequencing. If a dideoxynucleotide is inserted, remember, it stops the action of DNA polymerase from extending it. So we continue, if we look at this part, step 3, the DDNTP stops the reaction. So in other words, we start here, the first T that goes in is a normal, the G is normal, the A is normal, the G is normal, but by chance, in this particular reaction, a dideoxy T is inserted. And so what that means is it's going to terminate this particular fragment. So here would be the fragment that would correspond to termination at this particular point. Now you have to remember, there's the chance of termination at each of these nucleotides. So that's what's being shown here. So for example, on another strand, it may have terminated right there, which would have produced this fragment. On another strand, it would have terminated there, which would have produced this one. On another strand, it may have terminated there, which would have produced this one, and so on. So you can see that in that tube, we're going to be creating DNA molecules that differ in length by one base. And so then what's going to happen is you can use the process of electrophoresis to separate these fragments and couple it with fluorescence. In other words, each DDNTP fluoresces a particular color. And as these fragments move through the sequencer, going from smallest to largest, as the labeled nucleotide passes through, it'll fluoresce a particular color. And this is how automated DNA sequencing is done. You look at the pattern of the different colors that are produced, and you can determine what was the sequence of the complementary nucleotide strand. So, for other words, I can read this complementarity. It would be TGAGT, TGAGT. So in other words, what I'm figuring out here is the sequence of the complementary strand. If I know the sequence of the complementary strand, then by complementarity rules, G to C, A to T, and so on, I can figure out what was the sequence of the original strand. Now, you have to remember, the Human Genome Project involves sequencing 3 billion nucleotides, and there are all sorts of problems that can arise in the middle. But this idea of DNA sequencing was crucially important to allowing that particular project to take place.